Okay, I can check with him as well.
I will restart the session here. We're going to have two presentations now before opening our session on considering QACs as potential priority chemicals. So I want to introduce Keith Hostetler and John DeSesso, understanding that we might not have John on quite yet. Keith is a senior managing toxicologist in the Safe Bridge Regulatory and Life Sciences Group of Trinity Consultants. Prior to becoming a consultant, he spent more than 20 years in the specialty chemicals area with multinational expertise in toxicology and regulatory affairs. Keith holds a PhD in pharmacology and toxicology from the Medical College of Virginia at Virginia Commonwealth University. And John DeSesso is a principal at Exponent, Inc., which is a scientific and engineering consulting firm, and a professor at Georgetown University School of Medicine. He has over 4 decades of experience in the areas of developmental and reproductive toxicology, embryology, anatomy, and risk assessment. John earned his PhD in anatomy and teratology from the Medical College of Virginia, which is now Virginia Commonwealth University School of Medicine. So Keith and John will present on evaluating the safety of QACs.
Okay. Thanks to the group for inviting us to be part of this. I'm struggling now. I was expecting to see my slides closer to me or in front of me. Maybe I get that. Okay. So as I mentioned, we're going to be covering about 40 years worth of work here in the next 20 minutes. So we're going to have to move fast, and I'll turn it over to my colleague, John DeSesso, momentarily. We've already been talking about, through a number of presentations, about QACs, especially the disinfecting QACs and their important role being named and being included on EPA's list because of their efficacy against pathogenic organisms. They're unique and favorable in that they perform at very low concentrations, and when used as directed, over the years they've proven to be safe and effective when they are used as directed. They're registered around the globe for a number of uses, and that's based on the extremely robust data sets that have been developed over the years and reviewed by the authorities. We talk a little bit about a couple of different ways to use these. I have a slide here to just sort of focus in on 2 user groups. First, maybe the residential, the consumer side, where QACs are used and approved for use in food sanitizing or food contact surfaces, about a teaspoonful of a concentrate in 7.5 gallons, to give you a sense for how low they are and still effective. And more in a occupational setting, for example, in a healthcare setting, concentrations of 3,000 ppms, that's again diluting about a teaspoonful in 2 liters of water, still gives a very strong disinfecting healthcare grade product. In some of these cases, for the more concentrated uses, of course, personal protective equipment is indicated on the labels. As I mentioned, over 40 years, these compounds were introduced. I think we saw that from Dr. Arnold's presentation back in the 40s approximately, but they've been studied continually for that time by the regulatory authorities. They require far more stringent than peer review. They require guideline compliant studies that have to meet a range of demanding properties before they're accepted and can be used for human risk assessment. They require studies in relevant species that are predictive of human health endpoints. And when these reviews have been conducted, and they continue to be conducted on an ongoing basis, they've been shown both by US EPA and by the European Chemicals Agency to be approved for their current uses with no restrictions. Interestingly, that's part of what makes them unique as well, is that they're not systemic toxicants. They're not known to cause any adverse effect distant from where they actually touch tissue. If you give them orally, they cause gastric irritation. If they're applied dermally to test animals, they can be irritant, and in high enough concentration, even corrosive. But these are at high concentrations. The no observed or safe effect levels is what the EPA and ECHA use to ensure that the concentrations in the dilute products that people are using and potentially exposed to are safe. I'll highlight that Quats are not on any California Proposition 65 list. Dr. Arnold mentioned, and it has been well determined, that quats in the environmental fate are well understood. They bind to sediment predominantly. They do get removed from sewage treatment plants, and that improved with the Clean Water Act and improved wastewater treatment performance. They do not volatilize. These are non-volatile substances. They do not volatilize into the air. That's a fact. It's known. It's unquestioned. There has been tests on dozens of species of aquatic, both chronic and acute data. All that's taken into account when conclusions are drawn about ongoing safe use. I listed here the criteria for priorities that was discussed at the beginning of today, and I note some general observations. US EPA, from an exposure potential, estimates that unintentional but possible dietary exposure is less than a half a milligram, less than a quarter of a milligram per kilogram body weight per day. Dr. Salamova, from one of her publications, talked about dust, has quantified dust and estimated dust ingestion of far even below that. And we see from Dr. Arnold's presentation, low concentrations, micrograms per liter in wastewater treatment fluids. QACs are non-carcinogenic. They're not developmental reproductive toxins. Dr. DeSesso will cover that endpoint in some detail. Where concentrates are handled, personal protective equipment is required. They can be detected, as we've spent a lot of time too talking about detection. I will point out that there can be as many as several hundred similar compounds with similar molecular weights, and these have to be very carefully validated methods when it comes time to quantify. So they're not, they don't seem to be similar to other products or other substances that are listed on your priority monitoring list. I mentioned too, Dr. Arnold's work was very well received. We agree and believe that that's a fair representation. You can detect them in sediment. They're not detectable in surface waters. They don't migrate into drinking water. They have been tested. They're typically found in the parts per million, parts per billion, or lower concentrations in the environment, and these are well below any concern levels for effects on species. In terms of exposure, I think there was a question about exposure patterns. I would invite the panel to spend some time with EPA's 2017 work plans for ADVAC and DDAC. These spend quite a bit of time detailing exposure patterns, how people could be exposed to these substances. And in that, EPA identifies an acceptable daily ingestion of less than a half a milligram per kilogram per day. From Dr. Salamova's toddler estimation in her paper, this fraction of an exposure is some 700 times lower than that. Keeping in mind, this is a safe dietary exposure level based on all the cumulative robust data that's been submitted. Again, calling for research that there's urgency around risks associated with increased exposure when we're still talking about parts per million in dust seems a bit unwarranted. So the overall evaluation has been considerable. There's lots known about human exposure potential. There's tons of data of well-designed guideline studies and identified no risk levels in safe uses, and these are approved and used around the world because of this robust data package. Time doesn't permit me to go through all of the endpoints from a recent ECHA review, but this is an example. So in the appendix, the slides that you all have, you can see all of the endpoints that were looked at by the European Chemicals Agency. I highlight developmental toxicity because that's an element and a potential endpoint that has garnered some attention. Because of that, an expert panel has been convened to conduct a more rigorous and systematic review of both published and unpublished results. Dr. DeSesso chairs that expert panel, and if he's available to talk, he's going to take over the presentation from here.
Well, thank you very much. Can you hear me?
Yes.
Yeah, okay, fine. Thank you very much, Dr. Hostetler. So the recently, over the past 6 or 7 years, a series of papers have been published which allege that ADDAC and DDAC cause reduced fertility and neural tube defects in rats and mice. And as Dr. Hostetler has mentioned to you, these two chemicals have been used for over 50 years, and they've been evaluated by multiple regulatory agencies and considered to be safe when used as directed. So as a consequence, a teratology working group was impaneled to assess all of the developmental and reproductive data for these two chemicals and to ascertain whether these substances actually do cause developmental or reproductive effects. Next slide, please. So in order to do this, the panel elected to do what's called systematic review. And for those of you who don't know what a systematic review is, briefly, it's an attempt to objectively assess the scientific evidence to a clearly formulated question, which in this case is, do these chemicals cause reproductive or developmental toxicity? And it uses an explicit and transparent, well-defined methodology to do this. These are used both for the identification of the papers and reports to be reviewed, as well as selecting the appropriate ones to analyze, and then to critically appraise them and use only the most relevant research to draw conclusions. Next slide, please. So what we did was we did an automated literature review of the worldwide literature for using the names of the chemicals, ADDAC, DDAC, as well as benzalkonium chloride and their CAS numbers, and combined them with a variety of terms related to reproductive and developmental toxicity. And our search revealed about 789 potential articles. We reviewed the abstracts and titles of the culled list and came down to 8 in vivo laboratory studies that were performed in mammals. Next slide, please. Among those 8 studies, we found 2 chapters that came from a dissertation, and that dissertation was available online. When we looked at the dissertation, we found 2 additional chapters in that dissertation that had been written up in manuscript format, but had not yet been published. And so those are included in those 8 studies that we wanted to review. Now, in addition to the worldwide published literature, we also obtained safety studies for ADDAC and DDAC, which were supplied to the working group, and this constituted an additional 6 safety studies plus 4 dose range finding studies, which we sort of subsumed under the definitive studies that we received. Next slide, please. Now, in order to assess the quality of them and to be unbiased in the way we would do this, we used a software program called the ToxR tool method. It was developed by the European Center for Validation of Alternative Methods, that's ECVAM, and it was the methodology has been published on the open literature and is available on the net. And what this does is it asks a series of yes/no questions that you can answer either yes as a 1 or no as a 0. And the computer program keeps track of the yeses and nos, and it also keeps track of the topic areas in which these are where these are supposed to take place. And at the end of this thing, they evaluate each of the different topic areas and sum the scores, and categorize the studies into one of 3 categories. The best category, category 1, the studies are considered to be reliable without restrictions. Category 2, they're reliable, but there are some restrictions. And the third category as ones that are not reliable. Now, it's important to recognize that when we say the reliable on this, that this is for the purpose of assessing potential risk. All right, so are these good for risk assessment? Next slide, please. So the results of our ToxR tool analysis, we found 4 studies that were in category 1, 2 studies that were reliable with restrictions in category 2, and 8 studies that were considered not reliable. The 2 studies that were considered reliable with restrictions were among the unpublished studies. They were studies done in rabbits back in the early 1990s. And at the time they were performed, they met regulatory guidance. But recently, the number of rabbits that are to be used in studies has increased. And for that reason, we elected to downgrade those studies to category 2 because they don't meet modern standards. Next slide, please. So the big thing to consider, I guess, is to talk about the studies that were in category 3. And within that group of 8 studies, 5 of them are studies that have alleged that the reproductive and developmental effects of ADDAC and DDAC. And I want to discuss why they were considered to be not reliable. In general terms, there are numerous studies for risk assessment purposes with these studies. Many of the findings in the studies are told more as anecdotes than they are as experiments, so many of them don't have really rigorous scientific methods for the experiments. And oftentimes they use non-standard methods which were not really well described, and so it's difficult to understand what was done and how it was performed. So what I want to do next is, just as examples, talk about just 3 of the concerns so you get a flavor for why these things were different. And the 3 topics will include what the exposures were in the experimental conditions which used ambient exposures, how doses were calculated in those studies in which materials were given by diet, and to talk a little bit about the terminology used by the authors when they describe their findings. So the next slide, we'll talk about the ambient exposures. And the ambient exposure conditions, as provided by the authors, were animals that were in animal rooms in which a cleaning or disinfectant agent used a combination of ADVAC and DVAC in its formulation. In no case were either of these chemicals measured or identified in maternal tissues, that would be in blood, liver, or placenta, nor were they described or quantified in the embryos. And most importantly, the authors did not indicate what the mode of exposure was supposed to be. And this is interesting because those 2 substances have very low vapor pressures. This little table here indicates that compared to water, both of them are between 1 million to several billion-fold lower than water in terms of vapor pressure. So there's really not much of it escaping into the atmosphere for breathing purposes. The photograph in the lower right-hand corner is a photograph of the caging that was used in these studies, and you'll notice that they have lids on them. The lids have some holes at the top, or a hole at the top, at which a filter could be placed, but it's not a very large opening. And so the question remains, how do we think the animals got exposed to the material? I'd also call your attention to the fact that if you look at that cage, there are 5 mice in the cage. The cages are designed to hold 5 mice. The next slide. We'll talk about the dose calculations. And in this case, we recall that in toxicology experiments, dose is calculated as the mass of material that gets into an animal on a daily basis, or the milligrams per kilogram per day. In this particular set of experiments, the authors did not weigh the female mice during gestation, saying they didn't do that because the weights were variable. And of course, in mammals, they are variable, and mice gain approximately 40% more body weight during the course of gestation, which means that you need to adjust the dose as you go through gestation. We don't know how that was done in this study. And the dietary material was applied to the animals by a gel cube that had incorporated in it the laboratory cleaning solution. And these gel cubes were then placed in the cages on an as-needed basis. But the authors don't really tell you how they monitored how much the animals ate. A typical mouse in early gestation weighs between 30 and maybe 32 grams. It gets up to be as high as perhaps 35 or 38 by the end of gestation. So these cubes are pretty much the size, 80% the size of an animal. The authors stated that the animals consumed 28% of their weight per day, and it's not clear how that was measured because they didn't weigh the animals. And we don't know how they figured out how much of the cube they ate unless they weighed it before and after, but there were no records of that. And importantly, they weren't clear as to how many animals there were in each cage. And so if you had 5 mice in the cage, then you've got another variable there. Some animals might eat more than others, or some cages might only have 4 animals in it. So it appears to us that the doses were based on a series of estimates and not really on measurements. Next slide, please. And so the last thing I wanted to talk about with respect to this was the use of the term neural tube defects. And you have to understand that neural tube defects are really a loosely defined set of malformations that are seen at term. In humans, of course, that's after birth. In a mouse, that'd be somewhere around day 18 or 19 because gestation usually lasts about 20 days. These defects, neural tube defects, always affect the coverings around the central nervous system. That's the meninges that surround the brain and the spinal cord, and the skull or the vertebrae, which protect those. They may or may not involve malformations of the brain and spinal cord itself, and so that's an important difference. between what was described here and what we would see in a J10.5 mouse embryo. On the next slide, we want to point out that these embryos were identified and looked at on gestational day 10.5. Gestation period in a mouse is about 20 days, and the neural tube is a structure that basically forms something like rolling up a newspaper, and then eventually the two ends have to be folded together at the cranial end and the caudal end. And then that cranial end closes at about late on day 9 or early on day 10, which is just shortly before the time these authors looked at their embryos. And at that period of time, the meninges and the skull have not been formed, and they won't form for another 4 or 5 days. So they could not be affected. The photographs that they published show embryos, several of which, especially the treated embryos, are at a younger gestational stage than are the controls. And this indicates to us it's probably a case of developmental delay. And it is reinforced by the fact that when they did allow pregnancies to go to term, in none of their papers did they describe any neural tube defects in the offspring. So this suggests that the findings either were developmental delays, or they may have been findings related to animals or embryos that were dying, which is called a resorption. And resorptions occur quite commonly in rodents because rodents have anywhere from 14 to 20 embryos at one time, and they don't all make it through gestation. The next slide, please. Now, in terms of the other studies, our interim results looked at those other studies, the 4 in the category in the reliable and the 2 in the reliable with restrictions. And it's interesting that in none of those studies did we see any neural tube defects or any other malformations at doses as high as 200 milligrams per kilogram per day. Interestingly, the United States Environmental Protection Agency and the European Chemicals Agency have approved both ADVAC and DDAC. And when you look at their assessments, they're based on studies, all of which are in those category 1 and category 2 studies, as we had talked about. The most recent of those was the one performed by ECHA, which occurred in 2020, and that specifically considered and rejected data from those studies in category 3. And there's a statement here that they made that the quats could be used, those quats could be used as disinfectants in animal facilities as long as they're used following directions. So at this point in the analysis, the working group concurs with the regulatory agencies. Next slide, please. The conclusions, the interim conclusions of our systematic review at this point are that the data that we reviewed in the categories 1 and 2 indicate that it's a rather robust and extensive set of data. We're working through other data. We're hoping to get some more data from Europe to add to this, looking at both the published and unpublished studies. But at this point, we would be concluding that neither of those chemicals are developmental, nor are they reproductive toxicants. And thus, our conclusions align with those of EPA and ECHA. And it is our intention to publish this assessment once we're finished with it. The next slide.
The next slide's back to me. Thank you for that, John. And just to wrap up, the conclusion that QAC, disinfectant QACs do play an important role in protecting human health. They control a wide variety of disease-causing organisms, effective at low concentrations. And I really want to point out that guidance and concerns about use and overuse and frequent use, the CDC guidelines and EPA's websites on the proper use of disinfectant chemistries is really quite extensive and really quite complete. And offers specific directions on safe use, and I think that's an important consideration here. And that's the end of our presentation.
Thank you very much to both of you. We have 15 minutes now for questions from and comments from both panelists and audience members. And panelists can ask questions by just raising your hand. You don't see everybody yet. Oliver has a question to start us off.
So if none of these compounds are volatile, how can it be that it has been measured in air?
You're referring to Dr. Salmanova's paper?
Yes, that is just what we heard today. I'm referring to what I heard today. And there were clearly detectable levels in air.
My understanding was she sampled for 3 weeks at a time before getting detectable levels. Is my understanding. Maybe I misunderstood.
She just, okay. So on the one hand, I hear that you argue that there's no data. And on the other hand, you say there's no need for data because everything is safe. And because it is not volatile. Now, what I've seen today is, it has been shown unequivocally now in 2 presentations today that it's been absorbed and regularly detected in humans all the time in blood and in other biofluids. And B, that it has been detected in air by passive sampling. Now, these are 2 things I did not know before coming into the meeting, where I think these are in conflict to, what we heard before. And that's, based on published data before, but that's the idea of science. And that's also the idea of, going forward is to acquire data that may be, giving us more insights.
Well, I think there's a possibility. Am I on? Can you hear me?
Yeah, you're on, John.
Yeah, I think one of the things that could happen is that there's a possibility that materials could be used and spritzed and allow aerosols to form, and the aerosols might be a vehicle by way which some of this could get into the animals. But in terms of the way these disinfectants are used, and according to the directions, these lab sand is one of the substances they use, is to be used and left on the material on the hard surface and left alone, because that's how this work does its antimicrobial work. At that point, there should be very.
So you say that these products are never sprayed, so they should not be allowed to be sprayed in a spray.
No, I'm not saying in terms of the animal one. There's the animal one.
And that spray means generating aerosols, right? Is that correct?
Yeah, let me comment on that. Dr. Fine, you're exactly correct. They're approved for use in sprays. Typically, those pump sprays have coarse particle sizes that are not inhalable.
So they are in the air but cannot be inhaled?
A particle of more than 50 or 100 nanometers cannot get into.
They're not respirable.
They are sprayed in aerosol, but they cannot be inhaled.
They're typically, if they're high enough particles, they're trapped in the upper respiratory tract. They don't get to the lungs.
And then they swallow.
So and then they are, when they are either through dust or in the air, and we are exposed, so then we should not see it in the blood. Is that correct? Is that an understanding now? So how come we have 2 different reports today with unequivocally been shown to be detected in very frequent in the human population, when there's, not supposed to be in contact normally?
We're not saying that there's zero exposure. Nothing in my presentation said anything about zero exposure. My presentation said that when exposures in animals identify safe levels, in estimates of human exposure, which can occur through dietary exposure, through hand to mouth, result in potential exposures that are below levels that should cause concern. 10 nanomolar in plasma that may be bound to plasma proteins. I would be interested to know what toxic endpoint is being driven by a 10 nanomolar concentration of a quaternary ammonium compound.
So we have data showing that these can be incorporated into mitochondrial membranes and disrupting the mitochondrial electron transport chain. So that's data from UC Davis that we have not published yet, but that would be a way how things can be harmful. So, we're not saying, this is not a discussion here about disallowing any use. It's about, do we want to know more? That's, I think, what our California EPA is doing here, and why we're discussing this. And with that, I'm shutting up.
We're not telling you that you shouldn't be monitoring. We're telling you that what's the safety data when we know humans are exposed at low concentrations of what the data set tells us. That was the.
No. You said you have animal data. You have reviewed animal data, not human data. That's a different thing, because we before also heard that rats are not humans, and that rats have quite a different turnover than humans. And I'm sorry, you know, we don't have data on humans.
I would add just one final point to this discussion of particle size, since there's a question of exposure here, is to return to my point about foggers, that sprays, as Keith said, or was it John? I'm not sure, you know, produce somewhere from like 40 to 60, I think, micron size particles, but foggers, I've seen evidence about 1 to 10 microns, and those are surely respirable. So
I believe everyone using foggers are directed to use personal protective equipment.
But what does that say about the air that remains in place, and in terms of people who come through subsequently and all of that? So I just think that's as long as we're talking about spraying, we should make sure we're covering all of the particle sizes that can be produced. Other questions, Tom?
Yeah, I just, so I want to kind of go back to the point that came up at the end, which is, you know, you reviewed a lot of studies and scored them and focused on I forgot how many, the ones that scored low and that suggested a premise or a hypothesis of toxicity. And then you worked very effectively to at least provide evidence to refute those studies, and then went to the other studies that suggested safety, but kind of made the argument, well, those are robust and good. But what concerns me in public health, we always work the other way around. We don't really start with a hypothesis that everything is safe, or work against. We really want to try to refute the hypothesis of safety, right? We want that to be the robust finding. And I guess what concerns me is the hypothesis or the statement we're working toward is safety for humans. I don't think you can have a robust conclusion that something is high confidence of safety for humans if there's no human data. The reverse of that is that if something is toxic to animals, it isn't necessarily true that it's toxic to humans, but it means that you're less comfortable making the assumption or operating on that premise, right? We always err on the side of trying to refute safety where it makes sense. So I think, you know, what we're stuck with still is the absence of human data. We're still stuck with that. I mean, we're stuck with the problem we would have if it were toxic in animals, but no human data. We would say, well, I mean, there's a lot of substances that are toxic to animals, but they're not toxic to human. But we kind of start with that premise. The other way around is something that's safe for animals is not always necessarily safe for humans. We might move with that as a working hypothesis, but would always struggle to learn more about it, not be happy with a very small number of studies in a few animals that seem to be good studies, but could always tomorrow be refuted by the study that isn't true, especially when there are some poorly done studies that suggest that the operating hypothesis is wrong. Sorry if that's a bit winded, but that's kind of, I mean, I'm a little concerned with this.
I don't know if there was a question or not exactly, but I would say this: the requirements that regulatory agencies, US EPA and ECHA and others around the world, make a very high bar for what data has to be conducted in order for them to evaluate that data and make a judgment on human risk assessment. And we're not asked to go out and test in humans. Some of the exposure data that this industry has paid for has been done in humans. There have been human exposure studies that pass human HSRVs. That data is publicly available. So we're not trying to dodge. We're not trying to say that there isn't exposure. We're saying that we agree with the regulatory authorities' opinions based on sound risk assessment properties that, based on the data that exists, these substances are safe for use as directed.
Right. Although these agencies have made mistakes in the past. So I mean, the other question I have is we're looking at a class of 800 chemicals, and the studies that you're talking about are for a couple that are in use as disinfectants. So I think they're a bit different. And even if we were had good data for safety of 2 or 3 compounds, I don't know if it says enough about a class of 800.
I want to offer Amina the chance to address a question that was raised about her work, and then Libin, I see that you're here.
Yes, thank you. It's a comment on the first question about the inhalation and the air concentrations. Yes, we did deploy the air samplers for 3 weeks, but that's a standard, 4 weeks actually, but that's a standard procedure for indoor air sampling using pop samplers. And we do it for other compound groups as well, and we never see these high levels actually. These were probably among the highest levels for indoor contaminants that we've seen in our analysis, and we work with different chemical groups, including different types of flame retardants, PCBs, pesticides, etc. So that's a comment on the inhalation. So some of these compounds are volatile, especially ATMAX, because they also have lower octanol air partition coefficients. And in regard to that, I did not present the data, but we do have some preliminary data also on outdoor air, and those samples were actually collected using the high volume samplers run for just 1 day. And in those sampling, we are able to differentiate between particle phase and vapor phase contaminants. And again, we see an interesting pattern where we see higher levels of more volatile ATMAX in vapor phase collected with XAD absorbent, and higher levels of BACs in particle phase collected with filters. The levels for outdoor air are lower than for indoor air, but that's a trend we see for almost all contaminants that we work with. But again, the levels are higher than for the more known contaminants, like again, flame retardants, PCBs, and some pesticides. So just wanted to comment on the air data.
Thank you. We just have a couple minutes left. Libin, if you have something quick.
So quickly, one thing to clarify on the neural tube defect study. I read those papers. The ambient exposure is basically the facility using the QACs as disinfectants. It doesn't necessarily they are applying directly on the cage, but they could be exposed through air, through animal facility, working people. I think there are routes they can be get exposed to those compounds because they exist in a facility. So another comment I want to have is that I do not agree that all the federal agencies think they are safe. In fact, FDA a few years back has actually called for additional safety data for antiseptic over-the-counter products. Here's their language, and look at from their announcement in 2016. They said, since the FDA began review of topical antiseptic in 1970s, many things have changed, including the frequency of use of some of these products, new technology that can detect them, and FDA's safety standards and scientific knowledge about the impact of widespread antiseptic usage. And FDA is particularly interested in gathering additional data on the long-term safety of daily repeated exposure to these ingredients by consumers and on the use of these products by certain population, including pregnant women and children, and for which topical absorption of the active ingredients may be important. So I just want to clarify this concept because, even though in the older documents by, EPA, that can be traced back 2006, but I think federal agencies now becoming more aware of the additional data that's needed for this kind of compounds.
Thank you. We need to move on now, but I want anyone who has additional questions or comments, we have significant time for discussion, and please do keep those, and I can get to you first when it's next time for discussion. I see Vina had a question, and I will also check in, Jenny also, so I will put you on a list, and then I will also check in with Elizabeth if there was anyone else waiting to speak. So we'll have plenty of chance to get to your comments. Please do remember them. Thank you very much to our presenters, John and Keith, for your presentation. And we're going to move on to our final session of the day that is considering QACs as potential priority chemicals. I want to reintroduce Shoba Iyer, who's our staff toxicologist in the Safer Alternatives Assessment and Biomonitoring section in OEHA. Shoba will present OEHA's document on QACs as potential priority chemicals for biomonitoring California, and she'll outline the options for the panel. And then we'll have a chance for questions, panel questions, that is, a chance for public comment, and then the panel will deliberate. So I'll turn it over to you, Shoba.
Great, thank you, Meg. Can you just confirm for me that you can see my slides in full screen mode again?
Yes, all is good.
Great, thank you. Okay, my presentation now will be an overview of OEHA's potential priority chemicals document on QACs, as well as other information relevant to the criteria for potential priority chemicals. The potential priority chemicals document builds on OEHA's 2020 potential designated chemicals document and OEHA's 2019 preliminary screening document on QACs. The PDFs of all three of these documents are posted on the Biomonitoring California website on the page for today's meeting. Here I'm showing you again the criteria for recommending priority chemicals. The SGP can recommend priority chemicals for biomonitoring in California from the list of designated chemicals. These criteria are the degree of potential exposure, the likelihood of a chemical being a carcinogen or toxicant, the limits of laboratory detection, and other criteria that the panel may agree to. And I'll remind you once more that these criteria are not joined by the term and the panel is not required to specify other criteria. Now, we heard about limits of laboratory detection for selected QACs in Lee Bin's and Amina's presentations today. So in this presentation, I'll cover some information relevant to the first two criteria shown here. There is significant potential for exposure to QACs. I'll cover some relevant highlights on this slide. Of the QACs I reviewed, the national production volume for 20 of them was over 100,000 pounds in 2015. Of these, 11 had production volume of over 1 million pounds. QACs are used in a wide variety of applications, including as antimicrobials and disinfectants, as we've heard. Approximately 46% of the disinfectants for coronavirus on US EPA's List N include QAC active ingredients. Detections have been reported in indoor air and dust samples, and we heard about these recent findings in Amina's presentation earlier today. QACs have been detected in other environmental media, including sediment, sludge, and wastewater treatment plant influent and effluent. And we heard about environmental detections in Bill's presentation this morning, including in sediment samples collected from the San Francisco Bay. According to a summary of market research available online, the global QACs market is forecasted to grow by over 60% from 2019 to 2027. There are possible health concerns associated with members of this chemical class. I'll cover some highlights here, and most of this information is drawn from OEHA's 2020 potential designated chemicals document. Some QACs are linked with dermal irritation. One example is Quaternium 15, which is used as a biocide, preservative, and surfactant in cosmetics and personal care products and in cleaning products. It's a formaldehyde-releasing preservative. Some QACs are linked with respiratory effects. The Association of Occupational and Environmental Clinics has identified some BACs and one DADMAC as asthmagens, which they define as a substance known to cause asthma, which is acquired de novo from a workplace exposure. Studies conducted among hospital staff, such as nurses and housekeeping staff, have reported that exposure to QAC containing disinfectants and cleaning products can be linked with work-related asthma. This topic was covered at the SGP meeting in March 2020, and in today's meeting, we've had some discussions about potential for increased exposure to these subpopulations. In addition, respiratory effects such as pulmonary inflammation have been observed in studies of rodents exposed to QACs. Diquat dibromide and paraquat dichloride are quaternary ammonium herbicides, and exposures to these QACs have been implicated in neurodegenerative diseases such as Parkinson's disease. In rodents exposed to some QACs, reproductive and developmental effects such as decreased fertility and neural tube defects have been reported. We heard a presentation on this topic from Terry Rubeck of the Edward Via College of Osteopathic Medicine in Virginia at the March 2020 SGP meeting. Other literature on this topic was summarized and/or cited in the potential designated and potential priority chemical documents. Some immunological effects have been reported in vivo studies, such as alterations in immune response genes and in antibody production in mice exposed to some QACs. Altered cellular function, such as inhibition of mitochondrial respiration, has been reported in vitro studies of some QACs. And I'll note that plasma membrane disruption is the general mechanism of action that makes QACs effective as preservatives, disinfectants, and biocides, so it makes sense that the mitochondrial membrane can also be impacted. In vitro studies of benzalkonium chlorides have found inhibition of cholesterol biosynthesis and altered lipid homeostasis, and we heard about some of this research in Li Binzhu's presentation at the March 2020 SGP meeting. There are some publications about the potential for antimicrobial resistance to QACs. Shown here are screenshots of some of the work that's been published on this topic within the last year. We included these publications in the references list of the potential priority chemicals document, and we touched on this topic in the potential designated chemicals document. Here again are the panel's options following their deliberations this afternoon. The panel can recommend the class quaternary ammonium compounds, or QACs, be added to the list of priority chemicals, defer consideration of QACs, or decide against adding QACs as priority chemicals. And I'm happy to take questions at this time.
Thank you so much, Shoba. Sorry, finding all the appropriate buttons.
Same here.
We have time for, we have just a few minutes here for clarifying questions only from the panel, and we'll have a substantive time for substantive discussion. So clarifying questions for Shoba only now, please. Anything about the process of designating or considering these chemicals as priority? Carl.
Just like clarification on the listing as a priority chemical, I take it that that does not commit anybody to do anything, but it opens up the possibility of biomonitoring in case problems are seen. So in some sense, this is not a I don't want to understate it, it's not a particularly threatening thing. It just gives the biomonitoring committee and maybe some other agencies the possibilities of doing something with it if the data turns out. Is that
Is that correct?
Well, the panel recommended that QACs be added to the list of designated chemicals last March, which means that they could be biomonitored in a Biomonitoring California study from being on that list. Here, the panel has an opportunity to discuss recommending QACs as priority chemicals for biomonitoring.
Thank you.
We actually do have a couple more minutes because Shoba was quick. So Tom, go ahead.
Well, I just want to add to as I recall from previous discussions, we wanted the option to make these priority compounds because we got so much information on the rapidly increasing volume of production and use in our previous meetings. So that was one of the issues that motivated not letting them just sit in the background as compounds that could be brought forward at some point, but that there was sufficient evidence of rising use that it would be a good time. It would be a priority because this is similar to what we did to other classes of compounds like the cyclic siloxanes, where we said, oh, we want to get on top of things when the production levels are rising, not after they peak and then we start seeing them show up at levels that are a bit surprising.
Any other questions for Shoba? If there's no further questions, then we can open to public comment. We have about 15 minutes for public comment, and I'll just go straight to Elizabeth to see if there's anything submitted by email or by the GoToMeeting webinar interface.
There are no questions and no requests to speak at this time on Shoba's presentation.
Maybe I could just get clarification from Sarah. There's an open public comment that occurs at the end of the day. But Sarah, was this public comment meant to be restricted to Shoba's presentation?
No, this is not.
My understanding is. Okay, thank you. Sorry to interrupt. My understanding was this was meant to be more general.
That's right. This is public comment on QACs as potential priority chemicals generally. So it's the public's opportunity to comment on anything they heard earlier, anything they want the panel to hear before you guys discuss your recommendation. Also, depending on how much time you use in your discussion, the public could still potentially chime in during that discussion. But basically, now is the opportunity if anybody, either speakers or the audience or people listening from afar, want to chime in, this is the time to do it.
And we do have that unanswered comment from the last session. Sarah, if I may read it now, while we're waiting for other.
Sure. Yeah, I think we decided it was already covered, but if I'm not mistaken, but go ahead.
Okay. Well, this was related to a question that was answered, and I think maybe it was fully answered, but Emily Bryson from DPR asked, following up to the discussion that was the brief discussion Meg had about the QACs being applied via foggers, how would the use of a fogger affect the particle size and inhalation potential? And so that may have been sufficiently addressed, but that was the last remaining question we had.
Thank you for raising that. Just because I said it, I'll sort of mention again that the literature that I saw was that whereas sprayers can tend to produce larger particle sizes, like 40 to 60 microns, that fogging tends to produce particles that are 1 to 10 microns, which are, in the respirable range. But I would also welcome anybody else weighing in on that point, since it's just my looking at the literature, not having done that work myself. if there are speakers from other parts of the day who have done that related exposure work and want to weigh in. Otherwise, let's see if there's any additional public comment, which is open now for, as Sarah said, any topic from the day that you want the program or the panel to hear before our deliberations and vote on whether to recommend prioritizing QACs. I'll also just mention that Biomonitoring California received 3 written public comments last week. Those have been circulated to SGP members, and they're also posted on the meeting page for this meeting. One was from the AdBac Issues Steering Committee, which submitted comments regarding the use of analytical methodology for measuring antimicrobial QACs in environmental and biomonitoring samples. And there were 2 comments from Women's Voices for the Earth. One was about some special vulnerable populations that may have excessive exposures to QACs during COVID-19 and beyond. And the other was comments on the US FDA's review of AdBac and the identification of data gaps in the body of scientific literature on the safety of QACs. And I think that was actually referenced by Libin earlier. So just to highlight that those comments were received by the panel, and they're also posted for public viewing on the website for today's meeting. Elizabeth, any other public comments?
We have no public comments via email or via GoToWebinar at this time, and we have no requests to speak at this time.
In that case, I would suggest that we proceed to panel deliberation and recommendation, and I want to turn first to the panelists who were had something to say earlier that we didn't have time for, and that's first Vina and then Jenny.
Thank you. And Shoba actually mentioned this in her presentation as well. You know, in the previous discussion, several folks spoke about the lack of human data, and I had just wanted to comment on the strong evidence we do have on QACs and work-related asthma as one very relevant data set there.
Great, thank you. Jenny.
Hi, can you hear me? Oh, great. I'd like to thank the speakers for all the literature review that they did on these compounds. And I had a couple questions about the toxicology and risk assessment presentation. Just a couple clarifying questions. One was the study in the animal cages where they're using the compounds for like ambient cleaning in the room. What was the endpoint? I don't believe it was on the slide. I went back and looked. What was the reproductive endpoint for that study? Was it also neural tube defects, or what was the reproductive endpoint that they claimed in the study? That was one question. And that was my second question too, if you're going back to the slide. Sorry, go ahead.
Can I answer that question? Or I don't know if John's still on or not. That specific endpoint was the publication that was titled Ambient Exposure Causes Neural Tube Defects. So the endpoint was in fact neural tube defects, which was, using that term correctly, was incorrect.
So there was no other reproductive endpoint that was assessed in that study in the animal cages? I just want to make sure I'm clear about that.
John, I don't know if John's still on.
Can you hear me? Can you hear me?
Yeah.
Oh, yeah, okay. So there were some, the data showed an increase in resorptions in the highest dose, which was 120 milligrams per kilogram, according to what their calculations were. they were, I think they were, if I recall, they were around 12%, and the controls were something like around 6%. So they were, it wasn't dramatically increased, but it was increased.
Thank you. And then the other question was, you're saying for ingestion or for exposure to a toddler, and I just wanted to clarify, was that done on ingestion of dust only rather than any thermal or inhalation component, as some kind of more recent assessment of risks from dust have looked at exposures to the dust by ingestion, but also by resuspension of particles and inhalation and dermal exposure, and partitioning into air. And I'm just curious if that was based on ingestion of dust.
In my slide where I referenced Dr. Salamova's work, that was directly from her paper, which talked about dust ingestions and calculated exposure in toddlers on a milligram per kilogram basis. I do refer you to the work plan with EPA, where they look at all kinds of age groups and potential indirect exposure from indirect food contact, and therefore dietary exposure. And those have EPA's conservative modeling and what those dietary ingestions exposures are estimated to be.
I guess my last question was, I know you reviewed a lot of literature. Did you come across any effects on the microbiome of animals?
There's some of the chronic studies where animals were fed through their entire life, particularly in dog studies, because their microbiome was wiped out, then they got gastrointestinal distress and diarrhea from that. And that's not unexpected from an antimicrobial being given orally.
Okay, thank you very much for clarifying that.
You're welcome.
Lieben.
So I want to respond to Jenny's question on the, I guess, the study by Tara Ruback on the neurotube, and also some of the earlier study actually on reproduction. So I think in 2014, they published a paper where they carried on like a 6-month breeding study, like with exposure to quack. They found decreased reproduction, decreased fertility and fecundity in mice. So that was before the neurotube study. I think in neurotube study, like was a few years later, they look at the neurotube defects. And I think Keith or the other presenter also mentioned, even if there's no neurotube defects there at the later time point, but there's neurotube development delay, right? So there's that difference happening there. So, and another thing, you also asked about the microbiome. We did study look at those. I think my student has presented at a couple of occasions. We do see some alteration in microbiome. Exactly how does that affect, say, over longer term human health? We don't know yet, but the microbiome were changed with exposure. Yeah, and that's from one of the study where we actually analyzed the quacks and their metabolites in the feces. Yep.
Megan, there was, if I could, there was a point where Lieben was talking about the FDA's call for more data, and I was going to respond to that just when the commenting period ended. So is it appropriate? Can I make a comment on the FDA's request for more data?
Sure, if that was intended as public comment, and then we'll move on to panel deliberation.
Certainly public comment. FDA has in fact had quaternary ammonium compounds as antiseptic uses approved for over-the-counter use since the 1970s. Quats fall in a really unique position in that they were recommended by FDA along with isopropanol and ethanol during the pandemic because of the public health crisis of people needing to sanitize their hands. FDA has asked for new uses. These are new uses. They have asked for more data. The industry is cooperating, and they've actually done some exaggerated hand wash studies 30 times a day for 30 seconds and looked at what happens. And at the moment, preliminary data suggests concentrations are below any levels that the Center for Drug Evaluation has no concerns with the current data that's in hand. And it's being evaluated. It's a new use. FDA evaluates new uses differently than EPA. So that work is ongoing.
Thank you for that. I would note, as a physician, I can say that 30 times of hand washing a day is not exaggerated. If you're washing your hands before and after each patient and before and after meals and all of that, that is certainly not outside the realm of what you do on a daily basis.
So noted.
Just in terms of terming that study. All right, thank you all for your contributions. I just, as we move into the panel deliberation, one note is that Jose Suarez won't be voting on this item because he had to miss some parts of the presentations on QACs, but he will still participate in the discussion. So I want to open it up to panel members to discuss your thoughts and input to the program on the potential to designate QACs as potential priority chemicals. If anyone wants to start us off. Go ahead, Bina.
From my perspective, I think the QACs do meet several of the criteria for consideration as priority chemicals. The degree of potential exposure, as we heard about today, some concerning data on widespread exposures there, and on toxicity as well. We heard about in a previous panel meeting the work-related asthma data, as well as the emerging concerns on the reproductive and developmental toxicity.
Thank you, Jenny.
Hi, I want to agree with what Dr. Singla said, and also to say that we started out today by talking about focus for our biomonitoring efforts, and vulnerable populations and special populations in California were among those. And I think to me, the potential for occupational exposures, especially among some of the most vulnerable workers, such as cleaners, makes it like a very compelling story to me, and on top of the other considerations. And I don't see the use going down anytime soon. With the need to maintain safety and new variants coming up, I just feel the potential for exposure is perhaps only going to increase as people get out and about more to cleaned environments.
I had, Carl, I'll get you next. Just a similar thought to add on to what Jenny just said, that I see a lot of overlap between the people who are considered essential workers during the pandemic and those who are potentially more highly exposed because of increased disinfection use. And not just because they're, I mean, it's partly because they're out there working in settings that require, or at least are recommended to have increased disinfectant use. And I agree with Jenny that, you know, we could see this as a very temporary spike. And I think that's less likely than that some of these practices, or to some degree, they will become the norm. Especially when you see what happens to, we're seeing what happens to influenza cases and influenza deaths as a result of all of the, you know, masking and distancing and potentially also disinfecting practices that we've adopted during the pandemic. And it's reasonable to conclude that some of those practices would be recommended to continue even in the hopeful event that we could get the current pandemic under better control. So just to add to what Jenny was contributing, Carl.
Yeah, yes, thank you. I agree with Jenny and Megan, and I appreciated Tom's comment earlier about staying ahead of the curve, because we know that there are a lot of unfortunate substitutions that occur and it's very difficult for public health protections to keep up with the rapidity with which new chemical substances are created and used for similar purposes, and nobody has any idea how safe they are. And so it seems to me that all of this is an argument for making it a priority chemical.
Tom, and then Unha.
So I summarize it. I'm inclined to agree, but state this a little differently. I think, with regard to exposure and evidence of widespread exposure, I think the information we saw is significant and compelling for our case. I think in terms of the ability to biomonitor, what we've seen is compelling, so more than sufficient and compelling to go forward. And the only thing we might say that although the toxicity may be incomplete and not compelling, it is at a point where there are enough issues of concern to warrant action now. And it's kind of what Carl said about my point earlier. And to extend that is, when there is evidence of concern or sufficient evidence to raise concern, then it's in the best interest of public health to collect the data we need to understand that better. And that's one of the things we can do by making this a priority chemical. We will learn more, especially about highly exposed groups. And we will learn more to be able to sort them out into to see who's showing symptoms or not showing symptoms. But it's not the time to say, well, the evidence is not sufficiently compelling to declare this toxic. Well, the evidence is sufficiently compelling to say we could have a problem for us to move forward on the tools to understand the nature of that problem.
Unha.
I definitely agree with all the previous panel members' opinions. I think how we have plenty of examples of chemicals that, it came to so late once it's so spread it out in the environment, and then so many exposures are happened already. And then this we wait too long, and it's so hard to remediate and clean up and anything like that afterwards, or even we completely ignored, the high exposure groups. I think it's very important time, and it's critical because the public's understanding the use of disinfectants because of COVID-19, it's not going to disappear. It's going to be increasing, especially for the very young age children's exposure. It's going to be seems like more concerning. Not only those occupational high exposure groups. So I think it's very important to show the evidence, and a lot of environmental faiths and what we observed, and based on, it's convenient, and also it's very important based on the chemical properties and physical properties, that could predict, how those chemicals behave. But a lot of times the chemicals have a lot more, their behavior a lot more complex. So we cannot, there are so many examples that we say like, oh, these chemicals are not going to be persistent. These chemicals are not going to be in the air. There are so many examples we went through, but in the end, they were absolutely in the air. They're absolutely, persistent. So I think it's very important time I think we have to include them. We have to consider them. Yeah.
Jose, please.
Yeah, thank you for the time. I just remind that I'm not going to be voting on this, nonetheless, here are my comments. While looking at the criteria for recommending the priority chemicals that were listed there by Shoba on slide 2 and then earlier, it seems like for the limits of laboratory detection, we have ample evidence that these can be detected in blood, in stool, in urine. And so from that point, we have met that criteria. The degree for potential exposure, I think that's pretty obvious. That's been very well documented today. And then the likelihood of a chemical being a carcinogen or a toxicant. Here is, a little bit of the gray part of it, in the sense that the conclusion, well, at least based on my understanding, is that there is, to some extent, insufficient data really to show how toxic these chemicals are. But that doesn't mean that the lack of data doesn't mean that they're safe. It just means that there's a lack of sufficient evidence for this. So thinking of it more about from a precautionary principle type, even though this is way past that point, right? These chemicals were released back in the 30s and 40s. We need to reach, I truly believe that we do need to understand what's happening with this piece, especially given the large potential exposure and this very substantial increase in exposure that we've seen, especially this past year. And this is likely, this increase in exposure is going to be probably steady, if not increasing over the next few years. So from those points of view, I would be more inclined, even though I'm not voting, more inclined towards designating these as priority chemicals.
Thank you for that, Jose. You're by all means allowed to make whatever comments and contribute to the discussion as you like. Oliver.
Yes, so I was really intrigued today by seeing this, these two reports today, that we have frequent detection of these chemicals in various human biofluids. Before, when we started this a year ago, it was said that the idea or the overwhelming confidence of people in the field was that these would not be absorbed. And this is refuted. So this is very clearly not the case, because we can see it in humans. Now, we don't know the way of exposure. We don't know how they get into people, not for sure. There are multiple ways they could be done, but this was not tested on people with high exposures. So these were on regular students and regular folks in households, not even on people with high exposures. So that clearly justifies that we need to be knowing about the population at large at priority, because also the concentrations that were measured were higher, but sometimes a lot higher than for other chemicals that we otherwise monitor, right? At the same time, the chemical structure is made to be harmful to membranes. That's the idea, right? So, last time I checked, we have membranes. So I understand that nobody wants that these chemicals be eliminated from hospitals, right? Everybody understands the direct health effect of pathogens, and that we need to be able to have clean rooms, like in surgery, in a hospital. Absolutely, you don't want, infectious pathogens there. But that was the idea, and not, 30 times hand washing with that material and spraying in residential areas, and children cleaning classrooms. That was not the idea of these chemicals. That's not where, they were meant for. And I, in order to have a risk use balance assessment, we cannot just rely on data from rats, oral, given, because we are not rats and our metabolism is different. And it may turn out that when we have lots of data, that we see that in humans, the turnover is so high that the concentrations never reach high concentrations in tissues, in cells, in different organs. We may see that in 10 years from now, but today we don't have that data. And I think for California, seeing that high volume of these chemicals in various applications. And with exposure to people at risk, I think it's necessary to have this as a priority chemical on the list for the state of California. And we can only hope that the EPA will listen to our arguments.
So I am getting a clear sense that there's general agreement on the panel, and so I wanted to call for a motion, if anybody would like to make it, on the prospect of designating QACs as potential priority chemicals. Veena. So Veena, Dr. Singla then motions that the chemical class of quaternary ammonium compounds be included as priority chemicals for the biomonitoring program. Do we have a second?
Second.
So Carl, I heard that one first, seconds the motion, and then I'd just like to take a vote. I'll go through you as I see you on my screen, and you can indicate whether you support or oppose. Unha.
Yes.
I'm sorry. Yeah. What do you need? Voice vote or hands or what?
Carl, I took you as a second, so you're a voice vote already, and I'll request a voice vote of each individual person. Thank you very much. Okay. So Unha, I heard as a yes, and Jenny.
Yes. I support.
Tom.
Yes, I support.
Great, and Oliver.
Yes, I support.
And I also support this motion, so that makes it a unanimous motion among the voting members of the panel currently.
Meg, just to clarify, yes. Carl should still vote. You should still ask him for his vote. Seconding is seconding a motion. That's not his vote.
I support.
Thank you for that, Sarah.
Thanks, Sarah.
Thanks for clarifying. And now I can say it's a unanimous recommendation of the panel to include QACs as priority chemicals in the California Environmental Contaminant Biomonitoring Program. So with that, that concludes our deliberation and recommendation section, and we have 10 minutes designated for open public comment at this point. We'll accept comments on any topic related to biomonitoring California, and I will turn to Elizabeth for to find out if we have anybody who wishes to speak or has presented anything to the program via email or indicated they want to comment via GoToMeeting.
We do in fact have, we don't have questions yet, but we do have a hand raised. May I go ahead and unmute?
Hang on one sec, Elizabeth, because I see that Carl needs to say something.
Yes, thank you. This is my second meeting for the day, and I have a third one that began a few minutes ago. So if you would excuse me, I will leave. Thank you.
Great, thank you. Thanks, Carl. Elizabeth, thank you. You could go ahead.
Okay, Nancy Buermeyer would like to speak again, and I'm unmuting you now, Nancy.
Okay, can folks hear me? Great. Nancy Buermeyer, Breast Cancer Prevention Partners again. Unfortunately, I was not able to see most of today's schedule, but I did want to make a comment about the importance of what you're doing in putting chemicals on the priority list, and particularly classes of chemical on the priority list. The California Biomonitoring Priority List is part, as you know, of the DTSC Chemicals of Concern lists. And that list has been used in a number of pieces of legislation to get disclosure in product categories that we have not had disclosure in before. So cleaning products, the fragrance and flavors in personal care products, chemicals in menstrual products, and we're working on one now to try to get disclosure of chemicals in cookware. So every time you put something on that priority list, even if the program doesn't have the resources to do the actual biomonitoring, it still makes a huge difference in being able to better understand where these chemicals are in consumer products. And so I really want to thank you for doing all the work that you do and for continuing to add chemicals to this list. It's really important for the work that we do to try to advocate not only to know what's in these products, but to get them out of these products. So thanks very much.
Thank you for that comment. Elizabeth, anyone else who would like to make a public comment?
We have no raised hands and no questions submitted at this time.
Okay, then Veena has something to add.
Thank you, Meg. I just had a general comment or a suggestion that I think it would be helpful for the panel to ask all presenters to disclose funding sources for work presented to the panel and make explicit conflict of interest disclosure statements. I think it's those are very helpful in being able to evaluate any of the work presented to the panel.
I second that suggestion, and I just want to ask Sarah if there is a general sort of protocol that's provided to presenters in those lines. I know some people just take that practice when they're presenting their research is presenting the funding that they receive for that research, but has anybody given guidance about it?
No, actually, I think it's a great point, and I think Christy might still be on. Christy, are you available to comment on this? We haven't formally done this. I think I'm hearing Veena asking that we do this going forward. And so we're taking note of that, and not sure if Christy is still on it.
I am still on. Why don't we take this back as something to look at? And I understand what your comment is, Dr. Singla. So we'll give you a response and an answer, and we'll funnel that through Sarah.
Yeah, and we can address it publicly at the next meeting as well.
That's great. Thank you for that. Yeah, and I would also, well, it'll be dealt with publicly, but I very much support that request. Jenny had a contribution.
I was just going to further suggest that we follow a model for disclosure like the American Public Health Association does at their annual meetings, where you have a slide included in the slide deck that has to formally state that.
Or should we wait a little bit more?
There was a little bit of interference there, but as I understand, Jenny was recommending a particular model for that disclosure if the program decides to go that way, which is helpful. Jose, please.
Just a brief follow-up of that, and that usually comes at the beginning of the presentation, so people know from the beginning if there are any conflicts of interest.
Agreed.
Okay, so I want to check in one last time with Elizabeth, since according to the agenda, we're still in the time period for open public comment. Just make sure that others haven't indicated interest in speaking.
Not yet, and we're still checking the email. Last check of the Biomonitoring California email had none from that venue either. So it looks like we do not have public comments at this time.
And this is Sarah just confirming, yes, still no emails coming to the Biomonitoring California email. So yeah, I think you can go ahead and wrap up, Meg.
Great, thank you. So thank you all for the contributions to this meeting. Panelists are the visible ones, but the program staff does a tremendous amount to set up the meeting and invite the speakers and organize a really meaningful agenda for us. And the added difficulty of doing that all remotely is not lost on me. So I want to recognize the program staff and also everybody who joined us to present today, the public commenters, everybody who asked questions. It all enriches the conversation, and we appreciate your contributions. A transcript of this meeting will be posted on the Biomonitoring California website when it's available. And I think, as Sarah mentioned, the next SGP meeting will be July 16th, 2021, and will be held like this one via webinar. I want to thank everybody who contributed today and adjourn the meeting. Thank you.
Thank you.
Thank you, Meg.
Thank you.
Thank you. Bye, everybody.
Bye.